Dr. Sujit sir, almost it's 2:11. Nobody joined it.
I am also waiting, ma'am.
Yes, sir. What to do now?
Okay, let's wait for some time.
Dr. Bharati ma'am had joined. Ma'am, आपका like any presentation is there from your end?
No, ma'am. There is no presentation from my end today.
Dr. Bharati ma'am.
Okay.
Hello, ma'am.
Yes, ma'am.
I am audible.
Yes,
Yes,
ma'am.
ma'am,
You
you
are
are
audible.
audible.
Please give me a 15 minutes. I have in a lecture.
Okay, okay.
My presentation is ready, ma'am.
Okay, ma'am.
But
Okay.
after 15 minutes, I will join this
Okay, ma'am.
conference.
Okay, okay, ma'am.
Okay, thank you, ma'am.
Thank you. And Bharat Sharaf, are you ready for presentation?
Ma'am, yesterday only I presented.
Okay, okay. So let us wait some more time and anybody can speak a few words to not to make it to be because we had already started YouTube live streaming also for this session. Let us wait for 5 to 10 more minutes. Maybe somebody, they may join. Currently, we need to start. I think this 027 time we had given by 2:30, so they may join at the time. But all the time, whenever we are organizing such online mode, this is the thing is happening that nobody are taking that much serious. How we are going to attend the offline conferences itself, we need to take this off online also same because only thing physical mode and online mode, but everything is same. That the scrutiny of papers, since release of the brochure uh, till the certificates issue, everything we are doing in such a manner how we will conduct the offline mode. But I don't know why coming to this online mode, people are not that much serious. And here I wish to speak two things. One on which Dr. Sujit sir yesterday spoke with Dr. Rania ma'am. Like the habituated plants, whatever the plants they are growing, not exactly the place which they need to grow. Say suppose uh, Kashmiri apple here in Andhra, one place is there near Visakhapatnam district. The place is known as Lambasingi. Here, the people are thinking that almost the weather conditions are same as Kashmir, but not that much. But people had started grown up of apple and strawberries at that place. Initially, they went well, and some yield also they got. People got. Uh, but when we go for its taste or its composition, chemical composition of particular fruit, they are entirely different. So that is nothing but the habituation of the particular species, which we can go for GI also. If we come for GI, geographical indexes, that's why that has been termed as Kashmiri apple. If Kashmiri apple is grown up at Andhra, what will be the use that?
ते 4 ग्रुप आहेत पीपीटीचे ते एकत्र कसे घ्यायचे?
So this is what yesterday Sujit Jadhav sir had spoken with Dr. Rania Lampu ma'am also. Pavanjit ma'am,
Yes,
what may
ma'am.
be your opinion
Many,
on this?
ma'am, many times it happens when we are having, like, for example, nowadays we are altering these many species, right? For example, a few days ago only, I was going through the one video where what they are doing, they have mixed the stems of uh, strawberries as well as this guava. So now the guava is pink in color from the outside itself, or deep maroon color, and it is having the taste of strawberry plus guava. So this kind of things are happening, but the major issue will be there. For example, as you talk about Kashmiri apple, right? So the Kashmiri apple, it lost the essence now, because nowadays everybody is growing at their own place, right? Because of the weather condition, they are changing the weather condition so that they can grow such type of things at their home only. Earlier days, oyster mushrooms, they were not happening everywhere. Nowadays, as you can see, at any sort of climate, people are making the artificial climate and growing the oyster mushrooms anywhere, right? So technology becomes so advanced that people are nowadays doing these kind of things, but due to this, the originality, it is losing the essence. That is why it will be in future, it will cause issue in terms of copyrights and in terms of patent. Like earlier days, when these things were patented only on the name of a few countries, nowadays everybody is doing it, right? For example, this oxidized jewelry, like we Indians are wearing the oxidized jewelry since forever, right? But nowadays, at recent Lakme Fashion Week, what happened? Like at the red carpet itself, the celebrities or the models, they were wearing those jewelry by not saying that this is indigenous jewelry, but rather they have said, oh, this is a new kind of jewelry. But that was our patent, that was our thing only. But since we haven't got that patented on our country's name, so we lost it, right? So similarly, if such sort of things will be happening in case of agriculture, field or crops or any other thing, or even when we talk about the few fashion items, right? For example, few sort of saris, those were, or linen cloth, those were like we were wearing it since ages. But nowadays, those are into trend. But the Western countries, they are trending these by their own name, not by our own name. So if we are having such sort of technologies, we must get it patented the moment we are, if any sort of innovation we are doing, we must get it patented. That is what my intake of these things.
Yes, ma'am. Yes, exactly. Uh, Sometimes it is damaging the ecological balance also in my views.
Exactly, ma'am, exactly. It is damaging the ecological balance completely, right? Because the environmental conditioning which is required for the specific sort of vegetation or say horticulture, right?
Mhm.
It is required. But if we are dealing anything with the nature, then nature will definitely be playing with us after some time, right? Due to
Yes,
these
ma'am.
so many changes only, due to these many advancements in technology and making up these many roads, that is why we are losing the essence of, for example, in Uttarakhand nowadays, huge amount of vegetation and huge amount of forest are being cut, right? And
Yes.
that is how it will be facing landslides or flood in the future, definitely, because the trees who are holding the whole mountains, now those trees are going to be cut, right? And for that purpose, definitely there are landslides that is going to be happen. So how much we are playing with the nature, nature apparently will play with us. we need to think about this thing.
Yes, ma'am. Yes, ma'am. Exactly. The same
Right,
thing
ma'am.
we can apply in this year only. I'm observing that Amarnath Yatra. Have you heard about this, ma'am? This year, like within 4 days, that Shivaling has been melted, ma'am.
Yes, ma'am.
Uh,
Yes, ma'am.
yes, within
Due to
five,
the
within
huge
four.
climate change, it happened.
Yeah, yeah. So when we change, like we means the human tendency and human ideology and thinking, if we not aware of the ecological balance and environmental conditions, there may be very huge damage. But many technological innovations and inventions are going on. Many conferences and seminars like this are going on, but I don't know why
Yes, ma'am.
people are not
Ma'am,
aware.
actually, this all is happening, ma'am, because there is a huge gap. On academic level, we all are organizing the conferences and all. Industry people, they don't have any link with the academicians or the budding researchers, right? And the policy makers from the government, they are not connecting with these all, right? So the moment we will be having the multidisciplinary aspect where everybody, even the policy makers, the organizations, the industries, academicians, as well as the independent researchers, the moment they all will be coming together, definitely then only we will be talking about sustainability on reality. Otherwise, the same thing is going to happen because there is a huge research gap between the actual policymakers, between the actual environmentalists and the lawmakers, and the actual researchers which are working in this direction. So this gap need to be addressed.
Yes, ma'am, exactly. Uh, the same point, if we come for another angle, today morning, Dr. Kumar sir has been discussing that. Uh, in that talk, Dr. Sujit Jadhav sir had um, pointed out one point, like students are not able to aware of all these things, ma'am.
Yes,
Students are
ma'am.
not able to attend. In this aspect, I should appreciate you that you had encouraged your students very, very much, and I was surprised and I was very much happy of the work, being a very younger stage, like BTech first year, like that. They had done very good work and they are able to present also in such a conferences. So like that, every faculty member, not confined to chalk and talk only. If we are
Yes,
able to
ma'am.
encourage our students to do some societally useful work, and that has to be presented in such type of meetings, so as to inculcate many of the qualities in students, like soft skills
Yes,
and
ma'am.
presentation skills, everything we can inculcate.
Right.
But this awareness, first of all, the educationists and first of all the teachers like us need to get, to encourage students not only to get marks and get certificates, but
Yes,
also,
ma'am.
you know, in all dimensions. This is my opinion, ma'am.
Yes, ma'am. And one more thing, like we can inculcate this into preliminary education also.
Yes, ma'am. Exactly. Exactly.
For example, up to fifth standard, I agree that people can't be aware of science. We can't have science books up to fifth level, but up to fifth level, we can have at least few topics. For example, the social responsibilities, the waste management, environmental issues, right? These sort of subjects, if we are inculcating from first to fifth standard, it will be actually helping us a lot. At this starting itself, they know what environmental consequences are happening and how we can be able to resolve those issues. It would be really helpful because that is the only time when they are learning from their environment, right? So for that purpose, it is very important.
Yes, ma'am. Yes, exactly. Exactly. You're right. I request any other persons, any other participants can able to speak a few words.
How many presentations are left, ma'am?
Ma'am, actually, we need to start with number 27. Up to 35 are there, ma'am. Like
All right.
6 more presentations. Six
Okay.
or seven more presentations are there.
But
I don't
still,
know how many may
nobody
be
has
able to
joined.
present out of. Yeah. I think 230% need to join, but we had informed ma'am just before 20 minutes of the commencement time they need to join. But the same thing is happening,
Yes, ma'am.
the lack of seriousness.
Okay.
Okay. And maybe their own works also, somebody they may have classes at the time.
Right,
And
ma'am.
Dr. Neela ma'am, she had intimated me that she can able to join after 3:30 because her schedule is 3:20 to 3:30, but
Okay,
her
okay.
college timings will be up to 3:40, so that she had requested me to keep her presentation at last, ma'am.
All
That
right,
is
ma'am.
only I got. But remaining people, I don't know what is happening. I think Dr. Sadia Patel, you had joined, ma'am. Are you able to present your paper, ma'am?
Yes, ma'am. I am
Yeah.
audible, ma'am.
Yes,
Yes,
ma'am.
ma'am, you're audible.
Yes, ma'am. Please present your
Yes, ma'am.
paper.
Yes, ma'am. Wait 1 second. Good afternoon to all of you, ma'am. Is my
Yes, ma'am, your laptop screen is visible. You may start presenting your PPT.
Thank you, ma'am. Welcome to my PPT. And thank you for Sidvi Foundation to have me in this presentation, that is the multidisciplinary research and practice for sustainable development. Uh, my research work is related to the taxonomical fungi and
Ma'am,
from
your
this.
slides are not visible. Your laptop screen is visible, but not the slides.
Okay, ma'am. 1 minute. Huh?
You can share your whole screen so that whatever you will be opening, it will be visible to us.
Ma'am, just click on your PPT, ma'am, so that it will be open and it will be visible to us. Already your screen has shared. Just open the PPT.
Ma'am, you can present your entire screen. By that way, everything will be visible.
एक मिनिट 1 second है मॅम सारिका पाटील मॅडम सर मॅडम आहेत का जरा या की 1 मिनिट मॅडम पीपीटी जरा ओपन करा हो माझी सगळी स्क्रीन दिसायला लागले म्हणतायत पीपीटी दिसायला म्हणतायत लॅपटॉप दिसायला लागला पीपीटी दिसत नाही जरा बघा काहीच बोलत नाही. ते पीपीटी ओपन झाल्याशिवाय काय करतात? पीपीटी दिसना झाले, मी शेअर केले तर त्यांना स्टॉप शेअरिंग. खरं शेअरिंग होते की यू आर स्क्रीन शेअरिंग.
कनेक्शन नाही आहे अजून. काय ते नेट
पुअर कनेक्शन आहे.
हा करेक्ट.
मग सांग की
Connection
पुअर कनेक्शन आहे म्हणून सांगा मॅडम जरा. देर इज अ इंटरनेट प्रॉब्लेम म्हणून सांग. पण ते म्हणाले की शेअर करा म्हणून.
शेअर केलाय तुम्ही पण
पीपीटी नाहीये.
हा नेट
पीपीटी शेअर होणार झाले बघा जरा, पीपीटी शेअर व्हायला लागले तर खाली आहे की ग पीपीटी ओपन. ओपनच केलंय की.
स्क्रीन शेअर व्हायला.
स्क्रीन नको आहे ग. तुम्ही म्हणताय पीपीटी ओपन करा. ओके जरा नाही काहीच बोलत नाही त्यात.
Ma'am, firstly click on the stop sharing. Then
Acha.
you can again click on the share and share your entire screen. By that way, it will be visible. Yes, now click on the share and share the entire screen. At the top, there will be entire screen.
Entire screen. Okay.
Yes.
नाही मॅम म्हणतात एंटायर स्क्रीन.
In entire screen, whatever you will be opening, that will be visible to us.
कसं होतंय?
डिरेक्टली शेअर करा की.
मगाशी मी केलं होतं मॅम बघा.
स्क्रीन शेअर
दिसते का आता?
Visible. Just go to
Is it
Ah,
visible
yes,
ma'am?
ma'am. दिसते स्क्रीन.
हाँ ओके थैंक यू सर. हाँ
Ma'am, click
ma'am,
on
ठीक है.
F so that it will be full screen mode.
अच्छा ओके ma'am. Is it clear ma'am?
Yes, now it is visible. Yes, ma'am, it's clear.
Okay. Thank you.
Yes.
Sorry.
All right. Please continue.
Sorry ma'am.
It's okay, ma'am. It's okay.
Okay. My PhD research is related to the taxonomical fungi and especially on the black mildew. Then that during the explore of this black mildew fungi from Kolhapur district, I have a new species from new to science, that is the belong to the Tricasterina semicarpifoli, a new species from Asterinaceae from the Western Ghat, Maharashtra. And my abstract that is related to that species is it is a black mildew fungi belong to the order Asterinellans from the taxonomy of fungi, and we collected these new species from the Kolhapur district, Maharashtra, and based upon the detailed host specificity and morphological examination and comparison with the previously described taxa, the related species is new to the science, that is the Tricasterina semicarpifoli species nova. The present paper provided detailed description and illustration and a taxonomic of notes on the new species. Then my study area is a Kolhapur district, and it is a highly recognized as a biodiversity hotspot and belong to the northern western Ghat Sahyadri ranges. The Kolhapur district has a tropical monsoon climate, and this there are three distinct seasons: summer, winter, and rainy season. The period represent the peak season of flowering, fruiting, and sporulation in rainy season, and also that the diversity in the fungi, bryophyte, and pteridophyte also. So, the rainy seasons also support the emergence of black mildew fungi from Asterinaceae, then Meliolaceae, then the anamorphic genera like Asterostomes, Asterostoma, like that, making the Kolhapur and adjoining Western Ghat in an ideal region of taxonomical study of polycolous fungi and host plant diversity. Then the materials and methods, they include the black mildew infected specimens we are collected from a different localities of the Kolhapur district, especially from the sacred groves. These specimens were collected in a pre-sterilized polythene bags, brought to the laboratory and dried in a newspaper preserved in the standard size herbarium packets. Further, the host specimens are identified by using the standard flora, like flora of Kolhapur district from the Yadav sir and Sardesai sir, and by using the literature on the black mildew like Hosagoda sir and Hansford, we had identification of anamorphic where we described. Then the resultant discussion, the genus Trichasterina, specific, that is the leaf parasitic. Then the mycelium is ectophytic, Aprosoria laterals, mycelial and thyrothecial setae are present. The thyrothecia with the radiating cells, ostomatous and dehiscing stellately at the center. That is, they are dehisces in a star-like manner. Therefore, they are the Asterina. They belongs to the Asterina and the genus Trichasterina. Then Asai are globose, eight-spored and bitunicate, conglobate and two-celled. That one species which is known to science, that is the Trichasterina, and species is semi-carpifoli. The parent party species Nova. The microbank deposition is 84, 69, and 54. The entomology is based upon the specific host genus. Then the morphological characters are colonies are amphiphilous, black, dense, up to 6 meter diameter, hyphostrate to substrate, brown, opposite or irregular at the acute angles, closely reticulate, and cells have a 31 to 35 into 9 to 11 micron. The Aprosoria unicellular alternate unilateral brown, straight to slightly curved, and the mycelial setae. Presence of mycelial setae is a big character of this Trichasterina semicarpifolia. These are mycelial setae are scattered over the colony as simple. They are simple, straight, and obtuse. The thyrothecia are ovate to globose, and the scattered all over the leaf. The ascospore conglobulate, pale brown, and 30 to 33 into 20 to 22 micron. They are anamorph of this genera is absent totally. Then the specimen examined is on the living leaves of Alcidodacne semicarpifolia. Then the taxonomic notes, the Hosagodar and Goose described the Trichosterina from Tamil Nadu, and the Hosagodar et al. Described the Trichosterina from Kerala also. The present collection differs from the above specimen in a morphotaxonomical character on earlier record of this genus on the host family Lauraceae. Also based upon the host specificity, the present collection treated as a species new to science on unreported host of family Lauraceae. A new record of this species to the state of Maharashtra also. Then discussion, the Kolhapur district, extreme southern district of state of Maharashtra, which possesses high diversity according to 2,227 species belonging to 1,022 genera of 182 angiospermic families, Yadav and Sardesai. Therefore, the present research work is planned to investigate the polycolous black mildew microfungi from Kolhapur district. Before starting the present research work, primary survey of literature with the earlier record of black mildew was done. In present study, one species of black mildew fungi have been investigated, described, photographed, and investigated fungal taxa of black mildew fungi. One fungal taxa are found to be new on the new host, therefore belong to the host specificity, therefore the new record of respective fungal taxa. These are the some references, and these are the some photographs of Trichasterina semicarpifolia and the illustrations of this specimen. Thank you to all of you.
Thank you, ma'am. It's a great presentation.
Thank you.
Can you tell me, is there any current limitation of your research?
No, ma'am, there is no current any limitations of my research. These fungi, the specific role of these
Okay.
fungi is not yet known because these black mildew are not hazard to the plants, but they are maybe
Okay.
help to the plant. That is, the plants present in the sacred groves specific on that areas. These fungi are observed and not
Okay.
any polluted area, the black mildews are not observed. So I will
Okay.
also try to explore the specific role of these black mildew fungi,
All
specifically
right.
Asterina and Meliola.
Okay. Fine, fine. Thank you.
Thank you, ma'am. Okay.
Thank
Ma'am,
you.
you can stop sharing your screen.
Thank you, Dr. Bharti ma'am, for your wonderful presentation. I request anyone from the participant side who wish to interact with Dr. Bharti ma'am, it is welcome. And if any missed presentations are there here, presenters are there, it is welcome for their presentation.
Yes, one of the participant is with me. He will start his presentation.
Yes, sir. Yes, sir.
I call upon Mr. Popat Bhanudas Salunke, Assistant Professor, Department of Chemistry, Srimanta Bhaiyasaheb Rajamani Mahavidyalaya Maswad, and his number is 029, Statistical and Chemical Analysis of Mineral Content in Pomegranate Juice by the use of ICP-OES method.
Before that, I request Dr. Bharti ma'am, please stop sharing, ma'am. Or Rajshekhar sir, you can stop. Yes. Yes, sir. Yes, sir. The current presenter can share the screen.
How is it?
Yeah, it is visible. You may continue.
I am Professor Sarunke Popat Bandas, श्रीमंत भैयासाहेब राजमाने महाविद्यालय मसवड, तालुका डिस्ट्रिक्ट Satara. My name of the paper is Statistical and Chemical Analysis of Mineral Content in Pomegranate Juice by the use of ICP-OES method. Pomegranate means Punica grantum is considered as one of the oldest known edible fruits, and this fruit is cultivated in India or other countries about 4,000 years ago. Also, different fruits are cultivated, but this fruit is cultivated in India, in Maharashtra, and then in Pachim Maharashtra. Dry area, this pomegranate fruit is cultivated in large amount. Then in this fruit having scientifically different medicinal uses, that is for different disease, that is arteriosclerosis, cholesterol levels, cancer prevention. In this topic or paper, mineral composition of commercial pomegranate juice is determined. In this, clarification and filtration taking place, and then different pomegranate juice are given, and in this different minerals are formed. That is, for example, potassium, calcium, sodium, phosphorus, magnesium. In this, potassium having 183.40 mg per ml, calcium having 100.53 ml per mg per liter, sodium having 90.08 mg per liter, phosphorus having 70.05 mg per liter, magnesium having 72.25 mg per liter. So in this high mineral content of pomegranate juice contribute total intake of this constituent in human diet. Then this is the abstract of this chapter. Then keywords: pomegranate juice, mineral element, health benefit. Then next part, introduction part of this paper. In this recent technique, in this fruit is not only pleasant taste but also scientific evidence, and this fruit is used for in disease, that is reduction in blood pressure, prevention of prostate cancer, arthritis, anemia, diarrhea, inflammation, gynecological disease, different disease for this fruit is applied. Then also this fruit is popular drink in the Middle East and has marketed worldwide. Then in this fruit, composition of pomegranate juice depends on the cultivator type, environmental and post-harvest factor, and storage and processing factor. Then experimental procedure, and in this juice samples 25 pomegranate juice are formed, and in this 5 different varieties are taken, and in this 25 samples are taken, and then in this 100% pomegranate juice is given, and then 1 liter aseptic carton package are given, and in this juice samples were diluted with 1 with 0.2 percentage HNO3 and centrifuged for 20 minutes. Then standard solution were prepared by dilution of each pure element by nitric acid, and then used for mineralization of samples. All aqueous solutions and dilutions were prepared with ultra pure water, that is millipure Bedford EMA. Then result and discussion. So in this elemental concentration of samples are 100% pure, and in this different samples are given, and in this these are corrected with some values, that is element operating parameter for ICP OES, that is nebulization gas flow rate 0.55 liter per minute, auxiliary gas flow rate 0.2 liter per minute, plasma gas flow rate, then sample flow rate, operating power, Q, interface, sample uptake rate, spray chamber, nebulizer type, nebulizer setup, replicates. In this detection of fueling, that is standard values, that is phosphorus, sodium, potassium, calcium, magnesium, Fe, copper, zinc, manganese, nickel, lead, SN, cadmium, etc. In this, these standard values are given. Then next, in this sample is detected with minimum and maximum values, and then average standard values, that is deviations, that is phosphorus 30.07 to maximum 100.2, and then average value is 75.54 plus minus 20.55. These elements, different elements, that is potassium, sodium, calcium, magnesium, shows different values, that is minimum and maximum, and then average values. And then in this sample juice are detected, and in this juice different elements are present. In this series is given. That is concentration of various elements in the present work, that is decreasing order: potassium, calcium, sodium, phosphorus, magnesium, iron, manganese, tin, copper, zinc, nickel, and lead. So in this is the result and discussion. Then conclusion. In this conclusion, this pomegranate juice, that is pomegranate fruit, is cultivated in Maharashtra, in Pachim Maharashtra, in dry area. Larger amount of this fruit is cultivated. That is, this composition of fruit is depends upon cultivator, growing region, climate, fruit maturity, cultural practices, manufacturing systems. That is, in this pomegranate fruit, that is juice, in this, these fruit is prepared in Pachim Maharashtra in dry area, and then this quality of fruit is depends upon in this area, and large amount of this fruit is prepared and then transferred in out of country. Thank you.
Okay, thank you.
Thank you, sir. It was indeed a wonderful presentation. Sir, is there any fluctuation in the mineral nutrients, or is there any effect of solvent once you are doing the identification of mineral nutrients in the various juice samples?
Repeat.
Madam, please repeat the question, as we
Okay.
have lost the net connection. Please repeat
All
the
right,
question.
sir. All right. I said, is there any effect of different solvents when we are, analyzing the various juice samples? So, is there any fluctuations of the content or of the concentration, or even is there any a solvent effect? Hello. Sir, I was asking during the chemical analysis, when we are
Yes,
finding
yes,
out the
ma'am.
mineral
One
content.
second, please.
Okay.
No, no.
DMSO.
Hello.
Yes, sir.
Can you hear the answer he has given?
DMSO CDCl3.
Okay. All right, sir. All right. That's all from my side. Thank you.
Thank you, ma'am. Thank you, sir. Sir, anybody else from your end, sir? Dr. Sujit, sir, anybody else from your end to present?
Yes, yes. Myself, I'm going to present right now.
Dr. Rajendra Lavate, sir?
Okay, sir.
Yes, please, sir.
Hello.
Yes, please.
Okay, is my screen visible?
Yes, sir. It is visible.
Okay, so
Yes, sir. Your screen is visible.
yes, my topic is influence of boron on chlorophyll stability and photosynthetic pigments in sweet sorghum. As we well know, sweet sorghum is a drought-tolerant C4 crop, which has been used as a food fodder, fuel, and syrup. It has been used by these four Fs. And as our Maan Tehsil is semi-arid, dry climate conditions are present over here in Maan Tehsil. We have this jawar as a main crop, jawar and bajra as a main crops. And secondly, this jawar crop has been used as for high biomass and sugar accumulation, that is ethanol production. Nowadays we have one of the institution over here in the Fulton Tehsil, district Satara, where we have NARI, that is Nimkar Agricultural Research Foundation Research Center is there. Someone is disturbing, I think. Okay, so in Fulton Tehsil, district Satara, Maharashtra state, we have one institution, Nimkar Agricultural Research Institution. In short, we call it as a NARI. They have much more research on sweet sorghum, and they have developed one variety, the Madhura, what we call it as, and have worked on that Madhura variety. As far as boron is concerned, which is essential micronutrient, and it has been needed for the plant as they are useful in maintenance of cell wall and membrane stability. It supports chloroplast development, improves photosynthesis indirectly. Both deficiency and excess, they can reduce the growth. As we well know, boron is a microelement and it requires in very few ppm concentrations, which is naturally available in the soil. But if there is a high rainfall, that may cause some deficiency, may cause some effects on negative effects on the plant growth, as well as if the boron content has been in very less available for the plant, that may also cause some deficiency results. So my topic is that to develop to study the effects of different boron concentrations on chlorophyll stability, which has not been well documented yet. And it is need to identify optimum boron concentration, that is, which concentration is useful for the growth of economically important crop plant like jawar. In that also sweet sorghum what I have studied. And as I have already mentioned, Madhura is a variety that has been developed in Fulton Taluka, that is Nimkar Agricultural Research Institution, and that is why I have taken that crop uh, for the study. As far as objectives are concerned, to evaluate chlorophyll A, B, total chlorophyll, and carotenoids, these are my study areas where I have done the study. And as I have already mentioned, the another objective is to identify optimum boron dose, the dose which is helpful for the standard growth of the crop. Not an excess, not an minimum. The boundary between the excess and minimum availability of the boron is very poor boundary, and plants, they are accepting this boron at standard or optimum dose for their healthier growth. The hypothesis is that moderate boron enhances pigment stability, whereas excess boron reduces photosynthetic efficiency. So this is a structure of my work, complete random design CRD. I have used 3 replications of boron treatments that I have been given. The variety used is Madhura. And I have taken the observations after 45 days. It is because this jawar crop is of 120 days crop. And out of that, after 45 to 65 days, the reproductive growth has been started, that is flowering stage has been started. Before flowering stage, we have to study these photosynthetic pigments. It is because after 65 days, boron accumulates in the flower for the growth of the seeds, and that is why it may have some little effects in the leaves. That is, the presence of boron in the leaves may be reduced during the reproductive stage. That is why I have chosen 45 days for the this experiment. So seed selection, as I have already told, Madhura seeds that have been collected from Nari institution. Then pot preparation, I have made the pots with other pots that have been used. The organic fertilizers they have been used. Previously, the soil which is present in that pot, I have taken the pH and the boron concentration present in that original soil. Then sowing of the seeds, then boron application, then crop maintenance, then leaf collection after 45 days, and pigment extraction by Arnon treatment, that is in the 80% acetone. With the help of spectrophotometer, the data has been collected or analyzed. So this is the structure of my work. I have taken the absorbance at 663, 645, and 480 nanometers for measuring the photosynthetic pigments. We have taken 0.5 grams of fresh leaves, 80% acetone extraction has been done, then centrifugation and the ultraviolet visible spectrophotometer. Under this spectrophotometer, we have taken the readings for chlorophyll A, B, and total chlorophyll and carotenoids. So these are the treatments we have given. 0 ppm boron, that is control treatment. We call it as T0, TO. Then T1, that is 5 ppm boron. T2, 10 ppm boron. T3, 20 ppm boron. And T4, 50 ppm boron. Actually speaking, 50 ppm boron is excess to the plant, because whatever the study I have done regarding this boron, boron actually it is required in very small amount. But I have taken this particular 50 ppm boron concentration to study some hazardous effects of excess boron in the plants. So these are some results where we found maximum chlorophyll A, that is 0.35710 milligram per 100 grams of fresh weight of leaves was observed at 10 ppm boron. So here in the graph, you may observe the chlorophyll A. Similar results we found in the chlorophyll B also. Here we found chlorophyll B slightly higher than chlorophyll A, that is 0.65990 milligram per 100 gram fresh weights of leaves of Madhura plant occurred at 10 ppm boron. And as obviously total chlorophyll has been highest at 10 ppm, indicating optimum boron nutrition at 10 ppm. And as far as carotenoids are concerned, carotenoids are decreasing progressively with increasing boron concentration. Carotenoids, we do not found them higher than the control treatment. So as far as statistical analysis is concerned and the key findings are concerned, 10 ppm boron produce maximum chlorophyll A, B and total chlorophyll. 50 ppm boron concentration reduce the pigment concentration, and carotenoids, they declined with increasing boron concentration. So here we have done the ANOVA test for these 3 replications. And as far as discussion is concerned, as I've already discussed during the graph presentation, that is moderate boron, 10 ppm boron, enhanced chlorophyll accumulation. Boron supports chloroplast development, membrane stability, and carbohydrate transport. Higher boron, that is 50 ppm boron, reduced pigment concentration, suggesting excess boron may impair physiological processes. So findings are agree with previous reports on boron nutrition in different crop plants. So comparison with some previous studies, Goldbach and Wimmer in 2007, boron is essential for membrane stability. They also found the similar result, that is moderate boron concentration is required for the optimum or maximum growth of the plant. Han et al. In 2008, Shirin in 2018, they have already discussed that boron deficiency reduces photosynthesis or balanced boron improved chlorophyll. The present study, that is 10 ppm boron, gave maximum chlorophyll accumulation as studied in the previously by different scientists. So concluding this particular presentation, 10 ppm boron was the optimum treatment. Chlorophyll A, chlorophyll B and total chlorophyll increased under moderate boron, but carotenoids declined with increasing boron. So balanced boron nutrition is important for sweet sorghum physiology. As far as future scope is concerned, different field experiments should be done. I'm not saying that this much studies is important, because number of different enzymes related to carbohydrate metabolism or photosynthesis, they are having some role in all this physiology of plants. So study of antioxidant enzymes should be done, then measurement of biomass, sugar yield and chlorophyll fluorescence, that should be done for better results or for a particular result. So validate boron recommendations under different agroclimatic conditions. So as we well know, agroclimatic conditions, they may also affect or influence the physiology of the plant. So these are some future scopes for this experiment. This is acknowledgement and thanking you. I'm very much thankful for our principal who has given us permission to do this particular work in our Department of Botany. So thank you very much.
Thank you so much, Dr. Sujit. I do have a question. Like, if there is extreme weather condition, for example, prolonged drought condition, or say heavy rainfall condition, like extreme weather condition, so is it going to affect the boron release from organic matter?
Yes, sure. Prolonged conditions, it may be a winter, it may be a summer, or it may be a rainy. Prolonged conditions, they always affect the physiology of any plant. Not I'm talking about this jawar plant. Any plant, as we know well, in humans also, there are some different physiological effects we found due to the prolonged environmental conditions. Similarly, plants are also facing such conditions.
Fine, sir. And what do you think if we follow slow release boron fertilizers, will they be performing better rather than the traditional soluble borates like borax and all? The slow release boron fertilizers?
Slow release of boron fertilizers, we can use nanoparticles of boron. Now, one of my student is working on that. In Shivaji University, Kolhapur, we are preparing the
Okay.
nanoparticles of boron, and we are giving these particles from borate. We are preparing that, and we are giving that to the plant for the treatment, further treatment.
All right.
Instead of giving boron through the soil, it is better to spray the solution of boron on the plant.
All right. All right.
That has much better effects as compared to boron given through the soil.
Okay, sir. Thank you so much. That's all from my side. It was
Thank you.
indeed a wonderful and knowledgeable presentation. Thank you.
Thank you very much, ma'am.
Dr. Sujit sir, spraying on the plant maybe leave any residues, sir? Is there any possibility of leaving any residues?
No, ma'am. Boron is an essential microelement, first of all. And what in which days we are spraying it, that may, but there is no any report that boron is accumulating or boron is having residual effects.
Okay. Because this residual effect is very dangerous to the health of the soil. That's why I'm asking, sir. So
Yes,
if there is
but-
no residual effect, it is okay.
Huh.
Thank you so much, sir. That was a very
you.
wonderful presentation and wonderful work you are doing. Thanks a lot. Anybody else from your side, sir?
Yes, one more presentation is waiting for Dr. Abhishek Ranavre from our same from our college. Give me 5 minutes. I will call him up.
Yes.
No, ma'am, sir, I'm ready, sir.
Okay. Then you can start, sir. He's ready now. You can start, sir.
Good afternoon to all. My presentation topic is exploring the anti-cancer potential of cashew auricular leaf extract by using advanced chemical profiling like LCMS and study of the compounds which were identified through the LCMS and through the molecular docking for specifically for the mTOR protein and the certain gene expression analysis which we conducted here against osteosarcoma cell line, MG63 cell line we used. I will first start with explaining the LCMS analysis. When I conducted LCMS analysis, I found certain compounds which belongs to certain flavonoid, phenols, alkaloid, and their reported activities are reported through the right hand side. Means important antioxidant compounds like catechin, neocytosine, mangacillin, hyperin 6, they show very good amount of very good activity of as antioxidant activities, as well as quercetin 3 is also very well known antioxidant as well as anti-cancer properties. There are many literature available for their anti-cancer activities. Another is the camperol is also very good flavonoid, which shows the good anti-cancer activity. There are more than 33 different types of compounds which were identified, phytochemical or pharmacologically active ingredients identified through the LCMS. This is general overview of how phytochemicals act like anti-cancer. They induce the apoptosis, they activate the caspase cascade, they induce mitochondrial damage, and they arrest the cell cycle. Means this anyhow help to promote cell to becoming a malignant in case of tumor. For the proving our hypothesis, we conducted some cell cycle analysis. Here, flow cytometry analysis we used. It is based on the propydium iodide based iodide. Propydium iodide is considered as a dye which goes and binds to the DNA. And through cell cycle analysis, we found some interesting results, like when we treat our extract, methanolic leaf extract, to the MG60 cell line for 24 hours, we observed this type of results. Means around 80.30 cells are restricted to the G1 phase. And this is very good result, which we can correlate that the main aim of any cancer drug is to prevent cell going into the S phase. Means S phase we consider as a phase in which DNA synthesis is generally occurred. So our results show that we are getting good result of methanolic leaf extract of Cassia auriculata. Cells are staying into the G1 phase. This is the flow cytometry result of cell cycle analysis, which we conducted through the full flow cytometer. Propadium iodide was used as a DNA binding dye. Another interesting assay we conducted is a mitochondrial membrane potential assay. Mitochondrial membrane potential is a very good indicator to identify the whether cell is going into apoptosis or cell remain into as a normally. In case of cell, in case where a cell is going into the apoptosis or cell want to die, at that time there is a significant reduction in the mitochondrial membrane potential. And this is our means. Whenever we say that our drug is acting as a, or drug may act like a anti-cancer, there must be some physiological factors should contribute to the apoptosis. And one of the main important factor is the mitochondrial membrane potential. And we found that when we treat our extract to the MCC cells, we found that there is a decrease in the mitochondrial membrane potential compared with the control. This is about the decreased value of mitochondrial membrane potential. This assay is also done through the flow cytometer, and flow cytometer results at the bottom line we can see that there is a marked leftward shift in the fluorescence intensity, and this shows that there is a reduction in the mitochondrial membrane potential, and this reduction in mitochondrial membrane potential promotes cells going into the apoptosis, one of the important factor. Then we analyze certain genes expression, which we can say that important players for the during the apoptosis. They are like a caspase 8 protein is very important. Caspase 8 is considered as a pro-apoptotic protein. Means in our hypothesis was there must be increase in the certain genes which promote the cells into the apoptosis. We conducted one gel electrophoresis analysis. Here we basically cDNA technology was used. It means basically when we isolate the cells after the treatment with the drug and our extract, and we found out that we isolate the RNA. Through that RNA, we synthesize the complementary DNA of caspase 8, and that complementary DNA was analyzed using the gel electrophoresis. We found that one interesting result. We found that there is an increase in the expression of caspase 8. Means after the treatment of the drug, there is a chance that drugs cells are going into the apoptosis phase through the caspase 8 like protein. Caspase 8 is considered as a pro-apoptotic protein, means it takes up into the cells into the apoptosis. Another important protein which I identified through the gene expression research or the gel electrophoresis is the BCL-XL protein. Means at the BCL-XL protein is considered as an anti-apoptotic protein. Their expression should be decreased when there is a apoptosis is happening, and so we have 500 base pair is the gene. We can in the second lane is our methanolic leaf extract treated cells. We can find through visually we can find that the expression of the BCL-XL is reduced significantly, and this is the value. This is the image software were used to analyze the result, and we found this type of result. Another, when we found certain important pharmacological active compound through LCMS, we thought that there must be some in silico molecular docking, in silico studies should be done. For that, we use one mTOR kinase protein. mTOR kinase protein is very important. That this protein is targeted during the cancer. If there is some kind of mutation or proto-oncogene type mutation happens in mTOR kinase, the cell will not go into the apoptosis. Cell continuously try to grow, and there is a malignancy study. So the mTOR protein kinase is selected for the for the molecular docking studies, and for the molecular docking studies to analyze the binding of that certain phytochemicals. We found that we selected those compounds or those phytochemicals, which we obtained through our LCM study. We found that total 11 phytoconstituents we used for this analysis. Means any kind of enzymes or the protein, they generally having the binding sites. And the main aim of doing this molecular docking study is to understand the phytochemicals, how they are getting binded to the targeted mTOR protein. Mainly gamberin C was identified as the lead candidate. It binding around 9.0.59 kilocalorie per mole, forming the most exclusive hydrogen bond, and it shows more affinity. A common binding mode was shared by the flavonoids, including quercetin, camphorol. Even the quercetin, camphorol also they show the good binding affinity to the tryptophan is there to the glycine and valine residue. This finding indicate the potential for combining alkaloid flavonoid extract for the pronged effect on the osteocarcinoma cells, thereby reducing the probability of single target resistance. The mTOR inhibition signature is demonstrated to be consistent with the experimentally observed G1 arrest, reactive oxygen species upregulation, mitochondrial membrane collapse, means it is decreasing the mitochondrial membrane potential, upregulation of the caspase genes expression, and down regulation of BCL-XL protein. And this document results show good amount of means we are planning this taking this study as a in vivo when we are selecting some mice and through which we can continue our research. Thank you so much for giving me this opportunity to present myself. Thank you. Thank you.
Thank you, sir. It was indeed a wonderful and informative presentation. Sir, can you please go to the molecular docking slide?
Sorry, ma'am, I'm not able
Slide
to
number,
hear you.
I said, can you please go to the slide number 18, the molecular docking one? In silico
Yes, ma'am.
molecular docking. Yeah.
Yes, ma'am.
Okay. So, I wanted to ask actually with the only one protein you have find out the molecular docking study, or you have tried another proteins also?
No, only mTOR I targeted for my currently I'm targeting only mTOR protein, mTOR kinase.
Okay. So in future you will be studying studying
Yeah,
the
in future,
more. Okay.
my planning is to study more in the more details, more proteins, and
Okay.
mainly in vivo study through any live living model.
Right, sir. Because then there would be a complete, comparative analysis about the molecular docking studies. So it will be
Yes,
further
ma'am.
strengthening
Yes, ma'am.
it.
Yes, ma'am.
Yes. All
Yes.
right, sir. All right. That's all from my side. Okay. Thank you
Yes,
so
ma'am.
much, sir.
Thank you, sir. Thank you.
Thank you so much, sir, for your wonderful presentation. Uh, Sujit, sir, anybody else from your end, sir?
Yes, ma'am. One more participant is there. He's ready with his presentation. I call
Yes.
upon Mr. Anil Lam for his presentation.
Yes, sir, it's visible. Please continue with your presentation.
Yes. Yes, ma'am.
I said your screen is visible. Please continue.
Yes. Myself, Anil Marutilam from Srimanta Sahib Rajamani Mahavidyalaya, Maswad. Today my topic is the effects of herbicides on soil microorganisms, including bacteria, fungi, and actinomycetes. Across the globe, human activities, most notably our growing dependence on herbicides for managing agricultural pests, have sustainably reshaped both ecosystem and our cultivated landscapes. And because of these herbicides, number of soil microorganisms are damaged. Their role to increase the soil fertility as well as soil characteristics are decreased. Soil microorganisms comprising bacteria, fungi, and actinomycetes play a crucial role, and they maintain soil health, fertility, and the overall functionality of ecosystem. Then modern agriculture increasingly depends on herbicides to manage unwanted plants and pests, boosting how much crop produce. This increased use of herbicides raises real questions about how they affect soil's tiny inhabitants, which are vital for healthy soil, its fertility, and keeping the ecosystem in balance. Tiny inhabitants in the sense of various microorganisms like bacteria, they fix atmospheric, example in rhizobium, they lie on the leguminous plants roots and they increase the nitrogen in the soil, and therefore their inhabitants are damaged because of these herbicides. These soil microorganisms, including different types of bacteria, fungi, and actinomycetes, play crucial role in recycling nutrients, breaking down organic matter, and fighting off diseases, directly influencing how well crops grow. The central issue comes from growing evidence suggesting that herbicides can mess with the structure and functions of these microbial communities. This disruption can lead to a loss of biodiversity and changes in how microbes interact, potentially hurting soil health and the long-term viability of agriculture. Then in this chart, number of physical loss as well as soil fertility loss, loss of biodiversity, contamination, and carbon storage loss. All these occurred, means soil degradation occurs due to herbicides. Various types of the herbicides damage the soil health. Here, various types of the herbicides are here, like 2,4-D, as well as other various types of the glyphosate. All these herbicides damage the soil, and number of microorganisms are damaged, and they stop their role in the soil to increase the quality of the soil. Then, relative impact of herbicides on soil microbial communities. Here, glyphosate. Then next one is propazamide, and next one is fluitrol. All these affect the soil fertility as well as health of the soil. And the conclusion of this research thoroughly examines the widespread effects of herbicides application on soil microorganisms, specifically bacteria, fungi, and actinomycetes. It highlights how different herbicides influence these vital organisms. As evidence suggests, herbicides treatments so often lead to notable reduction in microbial diversity and richness. Chemicals like glyphosate, then propazamide, and fluitrol can have detrimental effects on soil health and ecosystem functions. The research problem centered on a need for comprehensive understanding revolve through rigorous experimentation, demonstrating that herbicide applications alters microbial communities and dynamics and impairs essential soil functions related to nutrient cycling and organic matter decomposition. The implication extend beyond just theoretical knowledge. They underscore potential long-term consequences for agricultural sustainability and soil fertility. Practically speaking, the study's results suggest a re-evaluation of current herbicide practices. It emphasizes the need for integrated pest management strategies that consider microbial health alongside crop productivity. Then in this chart also, number of types or their effects on microbial community are here, and all this damage the health of the soil, and therefore the soil quality as well as soil health day by day decreased because of highly use of these herbicides. Thank you.
Thank you, sir, for your presentation. Is there any limitation of the study or any research gap which you have found till now in the study?
Hello, please repeat.
I said, is there any research gap or any limitation of the study that we need to further take care of while going to the future directions in this research?
Actual sound.
Sir, I said, is there any current limitation of the study so that in future we need to take care of that limitation?
Yes, ma'am. In future, when we use herbicides in a low quantity or stop the using, then soil health increases or soil fertility maintained in future.
Okay, sir. All right, sir. Thank you so much.
Thank you. Thank you, ma'am.
Thank you, Dr. Anil, sir. Anybody else wish to interact with this presenter? Please welcome.
Anil sir, it is nice presentation. You can keep interest on these things, herbicides, health of the soil. We are supposed to talk about human being health and also the health of the soil. Thank you.
Yes, exactly, ma'am. It is more important as much as human health, soil health also that much important to sustain this nature.
Yes.
So, sir, anybody else from your end?
No, ma'am.
I request Nagesh, sir, are you ready with your presentation, sir? I request Dr. Nagesh Kale. Anybody else who left over their presentations at their time, it is welcome to present. Actually, here I could see Nagesh Kale, Magar. He is not responding.
Kevan Kothe sir is there, Rajendra Lavte sir is there. And
Yes, sir. Everybody, they are there in the meeting, but they are not responding, sir.
Nagesh
Only
Kele
Rajendra.
is there.
Anyone who's left for their presentation, they can present now so that we can easily bind up the session on time.
Yes,
Is there
yes, I
anyone
think.
who's left for the presentation? Okay. All right. Please, sir, present your screen.
Hello.
Yes, please, sir.
I think nobody is there, ma'am, to have the presentation.
Dr. Rajendra, sir, had unmuted. Dr. Sadia Patel, ma'am, are you able to present your presentation now?
Yes, ma'am. Good afternoon, ma'am. Just I'm sharing my screen, ma'am. Okay.
Yes, ma'am. Yes, please. Yes,
Okay.
please.
Give me 2 minutes only, ma'am. Just
Okay, ma'am.
Yes, please. Yes, please.
okay. सर मैं बस एक प्रेजेंटेशन देकर आई आप तब तो ये खोलो
नीरज शर्मा, ममता रामड़, सुशील कलारी, अशोक यादव, देवी नारायण, महेश निगम, रितेश मीडिया, ये रंगवा।
देखो ना सर मेरा 1 मिनट प्रेजेंटेशन में दे लूं एक देखो क्या लिखा है मैंने कितना लिखा है मैंने
स्टेबिलिटी तो मिल दी है, स्टेबिलिटी तो मिल दी है, मेरी आपका पूरा पे भी होगा दिसंबर तक, दिसंबर
तो आप ये देखो ना
किस दिया मैडम उन्होंने।
हाँ तो आप पांडे सर से बात करो मैं जब तक आ रही हूँ
इसकी फोटो खींच लूं क्या, क्या किसके आपको सेंड कर देता हूँ।
नहीं मुझे तो भेज दो उनको अपने से कोई प्रॉब्लम नहीं है उनको ये है
इसकी फोटो से लेना मैडम आगे पीछे की, ताकि आप लोग को नॉलेज में रहेगा, तो आपको मोबाइल से ले रहे हैं।
मैं अभी मीटिंग में हूँ 2 मिनट रुक जाओ भाई चलो मैं बस आ रही हूँ Good afternoon, everyone. Is it visible?
Ma'am, it is visible, but your voice is echo is coming. I think you had logged in with 2 devices. Please log out in 1 device, ma'am.
Is it okay, ma'am?
Now it is okay.
Okay. So, good afternoon, everyone. I'm Dr. Sadia Patel, working as an assistant professor in chemistry at Government College, Khategaon. It is situated in the district Dewas, Madhya Pradesh. So for today's presentation, I have chosen the topic Scientific Innovation for a Sustainable Future. We will talk about the advancing society, environment and economic growth. So first I want to congratulate the team of the organizers, those who have chosen this very critical issue and give us platform to discuss among you. So this one slide you can see, this is the current scenario, current situation of the earth. As you can see, we are facing the problem of the air pollution. We are facing the problem of the soil pollution as well as very important need of the human or other animals, water pollution. So you can see in air we have ammonia, carbon monoxide, carbon dioxide, nitrous oxide, methane, sulfur dioxide, and so many other harmful gases which are very, very harmful for the human beings as well as other living organisms. When we talk about the soil, it is also contaminated by the heavy metals and other food waste and oil. And water, we can see different types of bacteria, viruses, heavy metals, petroleum hydrocarbons are dissolved in water. So, in this presentation, we are going to discuss I'm going to discuss the nano remediation for all these critical issues, for all these problems. When we talk about the introduction, as a human well-being, we can use, or as a from the science background, we can use the science technology to improve the well-being of the humans. And what is the core idea behind it? When we take something, what is the nature of the human? We make something and after that we waste it. So we have to change this model into efficient, circular and low carbon model. It includes cleaner energy, smarter agriculture, better water systems, sustainable materials, and responsible use of AI and other data. UNESCO-style sustainability research also emphasizes that science should help reduce waste, improve efficiency, and develop more sustainable material. When we talk about the sustainability, I just want to give you one example. In MP, one very known town is there and very known city is there, Indore. You all might have heard about Indore. In Indore, that IT professional who was working in the multinational company, and before that maybe he was settled in the abroad, he came to Indore, and now they are living very simple life in the nature. They are using very less things to survive. So what is the whole idea behind it? We cannot change all the things at certain. We have to do all the things by slowly. So in that, we can see when we use very little amount of things, that means we are needing something very less. We have very less requirement, and when we have less requirement, we will waste less. So what we can do to see the solution of all these things, we reduce our requirement, we reduce our necessity. Okay, so this is a part of the sustainability. On the other hand, we know some requirements, some our needs are very essential and we cannot overlook it. So what we can do, we can use different technology to reduce the waste. So in that, nanotechnology is very important. In nanotechnology, we can talk about the nanomaterials. Nanomaterials are the materials which have very less size, across 10 to the power minus 9 to 10 to the power minus 12 meters. Or in that, what we can see on the first hand, you can see here, we can mix these all material with the fertilizer. What these materials can do, these can enhance the absorptivity of the fertilizers. We all know when we pour water into the crop, this water takes the fertilizer with it and brings to the water streams. So when we use the nanomaterial mixed with the fertilizers, it reduces the requirement of the fertilizer. It enhances the absorptivity of the fertilizer and it gives more time to fertilizer to be retained in the soil. So what is the benefit of it? As less amount of fertilizer is required for the crop to grow, and we see by the result of this, the pollution of water pollution also decreases. Without using the nanomaterial, when we see one example, we can see the pollution without the nanomaterial is more, and when we use the fertilizer with nanomaterial, the pollution percentage in water is very less. So this technology helps in agriculture to enhance the yield, crop yield, and it reduces the pollution. So this one technology we can see as a sustainability, as a core to reduce the pollution, and this is the scientific technology which we can use to enhance the Here you can see we have used the zero-agriculture as a green approach. In this you can see zeolite is the aluminosilicate which has porous structure, as we already have discussed. It reduces the leaching process of the fertilizer and enhances the absorption. This is the structure of the zeolite which we can use in the agriculture. As you can see, this is silica, it is tetrahedral structure. Alumina also you can see here. This is the schematic structure of the natural zeolite. It is taken by the GW physics Willig. Here one iron you can see ammonium ion, how it replaces the sodium plus iron, and this is the ion exchange of the ammonium ion for the sodium. This is the schematic diagram of the single application with the zeolite. You can see this is the theory of the zeolite application. So all you have seen, this is by the use of zeolite, which is an example of the nanomaterial, how we can increase the crop yield. This one slide I have taken from the Scopus journal, which shows the percentage of the article published in the scientific journal addressing the application of the nanoparticles in various diseases also. So here we can see the use of nanomaterial in the field of medicine. Here also nanomaterials are very useful for the medicine. See this one more slide you can see here, how we can improve the soil quality and how we can reduce the heavy metal mobility by the use of these nanomaterials. As a conclusion, we can discuss here, these soil solutions are the substances which improves the physical and chemical properties of the soil, which is beneficial for the crop production. Mineral fertilizers which are applied to agriculture do not have very great tendency to improve the yield, but they are very environment. These causes air, water pollution when applied at large scale and with large techniques. So application of the soil conditioner improves quality and heat of the crop reduces the harmful and negative effects of the fertilizer. Due to their unique structure and properties like iron speed and absorption. Natural purifiers are most appropriate in agricultural uses and they are environmental friendly too. So this is all about my presentation. I hope you all have understood about it. Any query? Hello.
Hello.
Yeah, ma'am.
Yes, ma'am. It was indeed a wonderful presentation. I have few questions. For example, when we talk about soil conditioners,
Yes,
so in the soil conditioners, it's very important to know about how much quantity of, whatever we are pouring there, whether we are applying the zeolites over there as a
Yes.
soil conditioner or even nanomaterials or other nano composites. So there are many other, even nano fertilizers are there. So when we are specifically applying the zeolites as a soil conditioner, what should be the maximum and minimum quantity so that we are not getting the excessive amount in the soil of those zeolites?
Yes, ma'am.
And what if we are we pour extra amount of zeolite? What will happen then?
Actually, as I have shown in my slide, these are actually aluminum silicate, kind of cement also. And we can say
Yes,
it's
ma'am.
like cement. So we cannot use it at the large scale. So what we did in my experiment, actually, this we have done it very small scale. I have put all these things in the pots, that pots in which I have planted the plant. So what we can see, we can see different amount we can take, like 100 grams we can take, 50 grams we can take, and we can mix the fertilizer. To all we use very less amount of fertilizer. And these zeolite increase the need of these fertilizers for the soil. We cannot use it at the large scale because these are very harmful for that plants also. It can cause damage to roots of the plant. So at the specific amount we have to use, and at very less amount we can use it for the betterment of the crops.
All right, ma'am. All right. Thank you.
Thank you so much, ma'am. Thank you.
Thank you, Dr. Sadia ma'am, for your wonderful presentation. Anybody from the participant side wish to interact with the presenter, it is welcome.
I think nobody is remained now.
Yes. Is Nagesh sir available for presentation?
Yes, yes.
Neela ma'am, after this presentation, you can present, ma'am. You can be ready meanwhile.
Yes, ma'am. Thank you, ma'am.
Yes, ma'am. Thank you, ma'am.
मैं रोज हूँ यहीं पे, थोड़ा लेट हो जा रही हूँ। बिल्कुल, हमने आपका इनविटेशन पा लिया।
Other participants, please
I
turn off
request
your
Dr. Sadia ma'am, please mute
microphone.
yourself.
Hello, is my slide visible to everyone?
No, sir. The earlier Yeah, now it's visible.
Okay,
Please continue.
my name is Nagesh Kele. I'm working as an assistant professor in Department of Chemistry at Shingney College, Sakarkeda. So today I'm going to discuss about engineering of graphitic carbon nitride. It's an photocatalyst. So engineering of graphitic carbon nitride based heterojunction photocatalyst. As in this photocatalyst, I'm going to embed Fe metal ion so that it can improve its activity. So the core argument will be parent pollutant disappearance is not enough. Practical value depends on mechanism, proof, toxicity reduction, catalyst, contaminant, and reactor ready metrics. So why this is important? The contaminant problem we are suffering in our India. Next one will be why we are using graphitic carbon nitride and the review contribution. So as we can see, our water is lots of polluted, and for that we need to use some photocatalysts so that we can degrade the pollutants from our water so that it will be pure and clean. So the graphitic carbon nitride, it is first of all pure. In the pure form we will take, it will be metal free, low cost polymeric semiconductor with visible light response. The band age can support oxygen reduction and oxidative pollutant transformation. And for this, the limitation will be recombination, modest surface area, and slow interface kinetics. So as we know, the fast degradation is not the same as deployable treatment technology. In the review contribution, we can see we will compare heterogeneous by mechanism and evidences, not just the rate constant K value. And connect materials designed to mineralization, toxicity, matrix, resistance, and recovery. So it will identify what is still missing before real water treatment deployment. Here you can see the review first. In the first emerging contaminants, the second one will be visible light photocatalyst, reactive oxygen species generations, and pollutant transformation. So first of all, in the first in the table or chart, we can see some antibiotics or pharmaceuticals or endocrine disruptors or any hormonal the molecule which will interrupt our hormonal imbalance, or pesticides we can say, or the dyes. So those are the toxic chemicals present in our day-to-day we can say water molecules or in the rivers or whatever the fresh water we are using. So what will happen? This photocatalyst, it will take visible light photocatalyst. Visible light, as we know, the range from 400 to 700 nanometer. Graphitic carbon nitride based heterojunctions, it will form the reactive oxygen species generation like O2 radical or OH radical or H plus ions. And what will happen is this pollutants or the antibiotics or pesticides, it will get destroyed, or we can say decomposed in the components like the cations or anions. The pollutant transformation will take place, and we can say the toxicity will be reduced, and we will get the mineralization like carbon dioxide. In the next slide, we can see the steps involved in this. The first one will be photon absorption, like first photon will be absorbed, then charge separation will take place. And as we have seen in the previous slide, the reactive oxygen species formation will take place, and then transformation. And after that, for the validation, we can check whether the water is pure or not by using kinetics or purity or toxicity, catalyst stability. In this, we can see different 5 types where we can see conduction bands and valence bands and semiconductors. How this type 2 heterojunctions and PN junctions, along with direct jet scheme, S scheme heterojunction, and Schottky assorted heterojunctions. In the first one, we can see that charge separation but weaker redox power as charge separation achieved but with reduced redox capability. And in the second one, we can see the p-type semiconductor as well as n-type semiconductor. So electrons are going from conduction band to n-type semiconductors of conduction band. After that, valence band of n-type to p-type. In the third one, we can see that strong electron and protons retain enhanced redox ability, which preserves strong redox carriers. After that, in S-scheme, we can see that band bending retains strong electron and holes. Band bending, we can see in the diagram as well. Band bending along with internal field retains strong electron and proton with redox capacity. The fifth one, Schottky assisted heterojunctions. Metal capture electron and suppress recombination. As now, the question arises, how will we synthesizing the graphitic carbon nitride? So the synthesis routes we can see over here. There are different types like thermal polymerization or hydrothermal or solvent growth, which is commonly used. In situ deposition, electrostatic self-assembly, microwave processing, thin film immobilization approaches.
Jesus, thank you, Lord.
Next one is interference and materials engineering levelers.
Jesus, thank
We can use 7 different types.
Thank God.
Exploitation of 2D contact, porosity control, heteroatom doping, valence,
Whatever
vacancy defect engineering, co-catalyst loading, morphology control, intimate interfacial contacts, we can see. Along with that resulting photocatalytic function, we can see 8 different O types, improved light harvesting, faster charge separation. In the faster charge separation, we can see efficient interfacial transfer will be there, like lower recombination and longer carrier lifetime will be there. Another one we can see stronger ROS generation in this.
Micro credential course. Okay.
Hello? Yes? Enhanced absorption, we can see higher mineralization, better stability. The stability will be better, like structural robustness, anti-photocorrosion, sustained activity. Along with that, easiest catalyst recovery and greater scale-up potential. We will be getting our catalyst recover after the pollutant degradation. As we can see, 3 columns like contaminating class, why it is difficult, why stronger studies must prove. As we have seen the previous slides, I'm showing you here antibiotics, endocrine disruptors, pesticides, and pharmaceuticals. So those are the toxic for us, and why it is difficult separation specific direct photocatalyst residue antimicrobial activities, pathways plus residue activity plus resistance relevant endpoints. So this is the reason why stronger studies must prove. Along with that, in the pesticides and herbicides, we can see that halogenated like fluorine, chlorine, bromine, or iodine intermediates and ion balance issues are there. So for that, dechlorination or defluorination products will be required. And in the pharmaceuticals, matrix competition, multiple functional groups are present in the pharmaceutical compounds, real water validation at relevant concentration. So in this slide, we can see contaminant specific degradation and safety evaluation funnel. We can see that this all 4 groups are merging at one place at absorption, specification, pH competitive sites. After that, it is getting into primary attack like selective oxidation. After that, the transformation like dealkylation, or if there is a ring, the ring will be get open. or if it is having fluorine or chlorine, the dechlorination or defluorination will take place in the third transformation. At the end points like TOC, CO2 ions residual activities. So the minimum evidence packages will be parent compound, transformation, mineralization, hazard, and catalyst safety. Catalyst as a photocatalyst, we will be using like graphitic carbon nitride. It will be retained after the all process. So this is the translation roadmap from catalyst discovery to water treatment development. First one, it will form a material design. After that, interface proof, like work function, XPS shifts. After that, it will go to intrinsic performance, environmental relevance, safety and durability, reactor integration, system assessment, and finally, pilot discussion. So, what are the review concludes? Graphitic carbon nitride heterojunction are promising because interfaces can improve charge separation and redox selectivity. As we have seen in the previous scheme, like type 2 PN Schottky, Z scheme, and S scheme systems must not be assigned from the band diagram alone. Mineralization, toxicity, reduction, and catalyst safety are the real treatment endpoints. So the future work should prioritize safe component, immobilized catalyst, and longer stability test, standardized photon flux reactor, and energy per order reporting. Real water validation at environmentally relevant concentration, techno-economic and life cycle assessment before scale-up claims. Thank you.
Thank you, sir. You have explained it so well. And all the depictions that you have used in this review article, those are up to the mark. So
Thank
I
you.
don't
Thank you
have
so
any
much.
specific questions from you. Thank you so
Thank
much.
you. Thank you.
Thank you, Nagesh sir, for your wonderful presentation. Anyone have any questions to Nagesh sir?
Yeah, ma'am. Good evening, Nagesh sir. Of course, you got the good compliments. But why did you include pesticides, herbicides in antibiotics column? Did you get my point?
Nagesh sir, ma'am is asking some question to you.
Hello.
Hello. Can you hear me now?
Yes, 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 yes.
No, it was, see, like I was looking at your slides. What I found out was herbicides and pesticides. And then they were included in antibiotics. ये क्या
Okay,
है?
like this, the compounds which will be used in our farms, like pesticides or herbicides, we will be using, no? So those compound will get mixed in water. So
Yeah.
after that, the peoples who are living in the villages, they will take the fresh water from wells or bore wells. So those we can see that the molecules we can degrade by using our graphitic carbon nitride. So that's why I kept in that.
Okay, okay. Nice. Good presentation.
Yeah, thank you.
Thank you, Nagesh sir, and congratulations to Prashant Mahalle sir. आपका guide है ना, Prashant sir?
Yes, 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 yes.
Yeah. Thank you so much.
Okay, thank you.
Now almost we came to the conclusion. Last presentation, last but not least, I'm waiting for this presentation because she is a very good speaker and she had delivered many guest lectures and keynotes for many conferences and seminars. We are fortunate enough that she had sent a paper to us, none another Dr. M. Neela ma'am. I request you to please present your paper, ma'am.
Thank you, ma'am. Thank you for the introduction, ma'am. Respected chairperson, distinguished guests, fellow researchers, participants, good evening, everyone. I'm Dr. M. Neela, Associate Professor, Department of Commerce, Kaveri College for Women, Tiruchirappalli. It's my privilege to present the research topic titled The Role of Human Capital to Attain Sustainable Economic Growth. I think since I belongs to Commerce Department, I have taken the title like this. As the famous saying goes, like the real wealth of the nation is determined by not only by its physical resources, but also by the quality, knowledge, and capabilities of its people. Yes, of course, this clearly shows the importance of why we have to invest in people, why it is essential for sustainable development. So, human capital is one of the most important and valuable assets of any nation. Unlike natural resources or physical infrastructure, human capital grows when people receive quality education and proper health care, skill development, and opportunities for innovation, lifelong learning. So, in today's knowledge-driven economy, countries no longer compete only through natural resources or industrial production. Instead, they can compete through the knowledge, creativity, innovation, and the capabilities of the people. So that is also very, very important. So sustainable development here that is focuses on the long-term economic development that improves the people living standards by protecting our environment, by ensuring the social equity. There are three important dimensions that contributes sustainable economic growth. One is environmental prosperity. Another one is social inclusion and the environmental sustainability. So these three dimensions contribute the maximum to the sustainable economic growth. Then economic growth plus social development and the environmental sustainability is important rather than it is increasing the GDP. So it is really helpful for both the present generation as well as future generations. Then these are the components of the human capital. Human capital consists of several important components. The education, first one is education. The education may be school level, college level, and skill development educations and professional degrees. Professional skills here means the technical skills, interpersonal relationship, the communication skills. So those kind of skills are needed for the human capital. And health here means the good health, that is physical fitness and mental well-being and resilience. So these are the things that is needed for human capital. Then research capability. Research capability also one of the important component of human capital. Digital literacy, they must aware about that is digital things. Then leadership, entrepreneurship creates the way of conducting the business. So these are the components that increase the labor productivity and competitiveness. So why human resource, that is human capital, is important here means the human capital plays a significant role in the nation's development. So it helps to increase the productivity because the skilled laborers, skilled workers perform tasks more efficiently than the unskilled workers. Then it promotes, that is, improves innovation. So by encouraging the research and development and the technological advancement, it helps to reduce the unemployment because it encourages innovation. Innovation means finding new things or search for new things. Innovation may be in the form of introduction of the new product, new service, or new method of production, new technology, whatever may be. Then it helps to increase the national income, thereby it enhances competitiveness. That is, human capital helps to improve the quality of life. It reduces unemployment through skill development and employability. So, moreover, human capital encourages entrepreneurship that leads to creation of new businesses and employment opportunities. Therefore, investment in education and healthcare should be viewed as an investment rather than the expenditure because the returns continue for generations and generations. Then human capital also contributes significantly to achieve the United Nations SDG, Sustainable Development Goals. Here, for example, SDG 4 promotes quality education. SDG 3 focuses on the good health and well-being. SDG 8 that is encourages decent work and economic growth, and SDG 9 supports that is innovation and infrastructure, industry innovation and infrastructure. SDG 10 aims to reduce the inequalities. So strengthening human capital directly supports sustainable and inclusive development across the nations. These are the drivers of the human capital development: education, healthcare, technology, and government policies. So these are the 4 drivers. The first is education. It provided through schools, colleges, and universities, as we all know that. Then the healthcare. The healthcare development can be attained through the nutrition, the preventive healthcare measures, and mental well-being. And the third one is technology. Now we are in the digitalized era, especially digital learning, artificial intelligence, and online education platforms. Then the fourth dimension driver is government policies. Here, the government also play important role by that is promoting schemes like Skill India, Digital India, National Education Policy 2020, UP 2020, and PM Kaushal Vikas Yojana.
It is good.
So next one is the human capital and the economic growth. Here, the investment in human capital directly contributes to the economic growth. Skilled workforce really increases the labor productivity, promotes innovation, it attracts foreign investment, and so on. Then there are some challenges in the human capital management. Despite we have the significant progress, there are several challenges also remains in this aspect. The first one is skill gap. These are difference between the skill that a job actually needs, but the worker skill is actually they carry. So that need to be fulfilled with the help of some training. Then brain drain. So brain drain also it means the highly skilled and educated and talented individuals moving from their home country to another country for the better living standards. Then poor quality education, digital divides, then unemployment prevailing in our country, then health inequalities, automation replacing our routine job. These are the challenges we are facing. In order to overcome these challenges, the government and educational institution can take necessary steps to improve the quality education. They can provide the lifelong learning, and they can strengthen vocational education, investment in healthcare sector. Then it can encourage research and innovations, and industry academia collaboration is much needed. Then digital awareness is needed. Women empowerment programs, they can offer more women empowerment schemes and programs. Then green skill educate, that is they can develop the green skills initiatives and empower the women through entrepreneurship. So human capital is the backbone of sustainable economic growth. So investment in people is most valuable. So countries that invest in education, healthcare, research, innovation, and skill development achieve their productivity. So in today's competitive world, the greatest investment in any nation can make is an investment in its people. So I would like to conclude with this statement: investing in people today creates a sustainable economy tomorrow. So thank you very much for your patience.
Thank you, ma'am, for talking about the sustainable growth overall, not only the scientific perspective, but you have, came up with the economic and social perspective also, which is indeed brilliant insight to this conference. Thank you so much. It was indeed a wonderful presentation.
Thank you so much, ma'am.
Neela ma'am, thank you so much for your wonderful presentation. I request anyone from the participant side wish to interact with Dr. Neela ma'am, it is welcome.
Yeah, good evening, Neela ma'am. Your presentation was good.
Good evening, ma'am.
Human capital has 2 things. One is knowledge, injecting, and other one is skills. So the economic side of the human being is nothing but a human capital. So, like how can we really improve our human capital? This is a very need of the hour, you know. So, can you have some tips? Do you have any tips apart from what we are doing in professional life? Of course, household life is there. But apart from this, any quick advice or quick things that we can really, what you call, build a human capital, you know, like from second floor to third floor or something like that. Do you have any tips?
Yes, ma'am. We can, that is, to improve the human capital, we can have more number of skill development programs in our curriculum. More skill development programs are important. Skill development programs can be, that is, it can be possible only through entrepreneurship. So entrepreneurship is the 21st century skills. So these skill development may be helpful for the students as well as the academicians. So they can, instead of concentrating on classroom education, they can revamp the curriculums and shift, that is, classroom learning from classroom learning to the digitalized learning, and AI collaboration can make it possible, and the critical thinking, that is trainings also helpful for them. Then human capital, that is, there are 4 important factors that required for any organization, ma'am, for producing anything. There is land, labor, capital under the organization. Land, labor, and capital, organization. These are the 4 important factors that required for the producing any product and services. Out of the 4 factors of production, human, that is labor, workers, that is human resource, is an most important asset, whether it is a large scale organization, small scale organization, medium scale organization. Human capital is very, very important. They can develop both, that is skill development through hard skills and soft skill development.
Yes.
Yes.
Thank you, and thank you, Andi Neela garu.
Thank you. Thank you.
Thank you, ma'am. Thank you, Neela ma'am. Thank you, Verisananda ma'am.
Thank you.
Thank you. So that's all for the 2-day international conference presentations. So I request Dr. Sujit Jadhav sir, anything else from your end? If not, we can go for the vote of thanks, sir.
Yes, madam. No any presentation from our side. So directly we will conclude this 2 days conference. Is it okay?
Yes, sir. Yes, sir. Yes, sir. We can go for vote of thanks, sir.
Okay. First of all, I will conclude and then I will go for vote of thanks.
Yes, sir. Yes, sir.
Okay. So regarding this conference, respected all the chief guests, distinguished speakers, honorable chairpersons, award winners, delegates, faculty members, researchers, students. As we conclude the 2 days international conference on multidisciplinary innovations and research for progressive sustainable development, ICMIRPSD 2026, it is my great honor and privilege to express my sincere gratitude to everyone who contributed to the remarkable success of this international event. I extend my heartfelt thanks to our esteemed chief guests and dignitaries, Professor A. Srinivasa Rao from USA, Dr. Dixon Adam from Ghana, Dr. Rania Lampo from Greece, Mr. Nagababu Abbaraju, Honorable Srimanta Ramrajan Naik Nimbalkar, former President, Maharashtra Legislative Assembly, Maharashtra, Honorable Srimanta Sanjeev Rajan Naik Nimbalkar, Secretary, Fulton Education Society, Fulton, and Honorable Srimant Raghunath Rajanaik Nimbalkar, Chairman, Governing Council, Fulton Education Society, for their inspiring presence, valuable guidance, and unwavering support. Their insightful thoughts have truly enriched this conference. I also express my sincere appreciation to our principal, Professor Dr. V.C. Kakade sir. In very short period of time, he has granted us the permission to organize and arrange such an event. And due to his consistent support, we are able to run this particular 2 days conference very successfully. I am very much thankful to him. I also express my sincere appreciation to our keynote and invited speakers, distinguished chairpersons, co-chairpersons, reviewers, and all the researchers who presented their valuable scientific work. The exchange of innovative ideas across diverse disciplines has reflected the true spirit of multidisciplinary research and sustainable development. I congratulate all the recipients of the Sidvi Special Awards, especially Mr. Chinmay Prakash Sawant, who is the recipient of the Best Environmentalist Award 2026, Mrs. Mahalakshmi B.R., recipient of Research Excellence Award, and the recipient of the Young Researchers Award, Mrs. N. Sai Prashanti, Mr. Amol Rajendra Singh Pardeshi, and Dr. D.S. Wadavkar, sir. Your achievements inspire the entire research community and encourage young scholars to pursue excellence. I express my sincere gratitude to the management of Fulton Education Society, entire governing council, as well as the college local management committee, especially with reference to Advocate Srimanta Prithviraj Rajamani, who is also encouraging us for having such events in the college. Then I sincerely express my gratitude to the Sidvi Foundation, without whom, and the Sidvi Publication House, without whom this event is not possible at all. The IJSEM editorial team and the entire organizing committee, all the faculty members, especially our convener, Khatal sir, and coordinator More sir, all the other faculty members from science faculty as well as IQSC of Srimant Bhaiya Sahib Rajamani Mahavidyalaya Maswad, as well as some student volunteers and technical support team for their tireless efforts in making this conference a memorable success. Finally, I thank every participant who joined us, both offline and online mode, for your enthusiastic participation and valuable contributions. I hope the knowledge shared, collaborations established, and friendships formed during these 2 days will continue to inspire impactful research and meaningful partnerships in the years ahead. On behalf of the organizing committee, I wish you all good health, continued academic excellence, and every success in your future endeavors. I am very much thankful to all of you, and I stop here with my concluding remark. So, concludingly, I have already express my thanks to all the dignitaries who are there with us for having this 2 days international conference. Thank you very much, all of you, and to all the participants. Within 2 days, we will give the certificates on your registered mail ID. Send you the certificates on the registered mail ID. And afterwards, we will have Rashtrageet, and then we will stop this particular conference. So we are starting the Rashtrageet, national anthem.
Yes, sir. Thank you so much, sir. So I request all of you to please stand before your devices to join your voices for singing national anthem. Before that, only small thing I need to announce that as Sir said, we are about to send the certificates within couple of days to all the registered participants' mail IDs, and Very soon we are going to release our conference proceedings also. I'll be sharing in the group for the review. Review means any corrections like that. So first correction after first proof and second proof. So finally we'll made it to be published. So thank you so much for all of you. Now I request all of you to please join your voices for the national anthem.
Please start.
Yes, sir.
जनगण अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तव शुभ नाम जागे तव शुभ आशीष मांगे गावे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 भारत माता की जय
Jai, Jai Hind.
Thank you.
Now I'm officially concluding this meeting. I request Mr. Rashekar sir, please end the meeting to all.
Yes, madam.